The last thing I'll show you is lab resources. So what kind of support students get for labs. So with every labs, we give them how to do it videos. Basically, what you're doing in the lab. So videos you should watch before you come to the lab. Then we've got the manual, then we've got specific lab folders. So if I go to lab one buffers, there's quite a bit um, they can use. So we provide them with academic sources for their lab. So these are journal articles that will help them understand what their lab's about and how to answer some of the questions in the lab. So we provide these articles for students to use to read through, so it sort of supplements their lab reports. So it really builds in them the ability to use academic sources to synthesize a very strong argument, which is what will get them a lot of the marks. So we provide them with this. They don't have to search for any resources. I mean, we encourage them to do it because it's always good to search for your own resources, but we give them as a head start just to help them out a little bit. And if I go back, we've got academic sources for why theoretical calculation or approximate, again, another resource for them to use. If I go back here, there's a folder here, additional help with writing your lab reports. So there's a few things that a student can use here. There's a document that gives advice on how to write a lab report. So if I open this, it tells you how to format your tables properly, what's right in this right-hand column and what's wrong in the left-hand column. For example, lab reports, they should never use personal pronouns. They should always be in passive tense when they write lab reports. We've shown them how to correctly tabulate data, how to find academic sources, how to properly write a conclusion. So if I open up another document, here is one where it shows students how to carry out certain calculations when it comes to uncertainties. So it gives them many examples that students can replicate in their own lab reports. Then we've got a document which is actually a model lab report. So this is an actual real lab report. It's for an experiment that they don't do, but this is the actual format of a lab report the student should follow and emulate. So we've broken it down into the sections they should have in theirs in terms of a full answer, a full discussion, a full conclusion, and a full reference list. So a student can basically look at this and map it to their own work. So they know, have I got this bit? Have I got this bit? Have I got this bit? And then it's basically a checklist of, yes, I followed the structure correctly. This is what a model, this is what a lab report should look like. Does mine look like this? So this is the type of additional support we'll give students when it comes to writing their lab reports. And if they engage and use these, the lab reports tend to be very good. I mean, it's quite hard to go wrong after using some of these sources, to be fair. And of course, there's some other stuff with academic sources, which we give them because we always try to encourage them to search for their own sources. So these two documents, I'm not going to open them, but they basically show them how to search for academic sources, whether it's Google Scholar or other websites and things like that. So this is quite a sort of a basic introduction to the labs. So in our first term, the students have a cell biology module, and one of the first key skills they have to do or learn is how to properly focus and navigate and use a microscope. So prior to them attending a lab, particularly for students who've never done it before, this is something we'll ask them to do in their self-study. And it goes through all the key steps and details of how to use a microscope. So I'm just going to go through this. And the really key thing here is it gives students all the details of the main points. It gives them activities to do, and it also tells them when they've made a mistake in something they've done. So here it's just told me about the basic structure of a microscope and the fact that there's two dials here that controls the focus in a microscope. So a coarse one and a fine one, or you can call it the rough focus versus fine focus. So if I go to zoom, it will go into actually making me change the focus so I can magnify and get the resolution on an image. So up here, this sort of purple circle, that's my blurred image. So what the student is asked to do is focus this until we start to get a bit of an image. So this circle here is just a front-on view of this dial. So if I rotate this, you'll start to see the stages moving on the microscope, because I'm changing the resolution. So I'll keep going until I start to see some kind of image. So there I'm starting to see very blurry cells. But because it's only a 10 times magnification, to get a really good image, I have to go a bit further. So I'm going to just check it, and it's going to say the image is not quite in focus, so keep adjusting it. So if a student isn't quite there with what we're looking for, it'll ask you to retry. So what I'm going to do is I'm going to adjust it to fine focus, because that's what it's asking me to do, and then refocus it until I'm really clear. So now that image is really, really clear. It's high definition. So if I check now, it'll say, well done, you've perfectly focused the image. So now the student knows what exactly they're looking for when they're magnifying their first image. So then it'll ask us to go into the further magnification. So this is when I change from times 10 to times 40. And now it's going to ask us to refocus and get a closer look at the image. So I'm going to change to focus. Then I'm going to go through again, focusing it until I get a much more clearer picture, which I think is about, which is about there. Now you can see the picture looks a little bit dark. So there's an option here for brightness. So I can turn up the brightness so it becomes much more clearer for me to see. And again, these are the controls that the student will have on the microscope. Bottom left-hand side, they'll have the little dial that controls the brightness. So once we're happy with that, we can now start to see some cells. Okay, and the little black dot in the middle, of course, is the nucleus of our cells that the students are going to be looking at. So of course, we want to go even closer and try and see the cell even more closely. So I'm just going to check to make sure it's happy with me. And it says, yep, you've done everything well. So we can move on to the next bit. Now, before we magnify to 100, we've got to put an oil drop on our lens. And this has got to do with light and refractive index and that sort of stuff. So I'm just going to move the lens a little bit away, then use this bottle of oil to drop a small amount of oil onto the lens. And then if I put my 100 magnification on and check, it'll tell me that I've correctly dropped the oil in the right place. And now the lens is ready to look through. Without this, it'll cause some problems with our magnification, and it could even damage the lens without the oil. So we're now ready for our 100 times magnification. So as before, I'm just going to slowly rotate this until I've got a very clear image. I'm happy with that. I'll adjust the light again, just so it looks much more clearer. And I'm happy with my final picture. And I can see there a group of cells with a nucleus, typically what the student will see in the real life lab when they're doing their first module. So if I just carry on through, I'll just check it to make sure I've done it correctly. It'll go through some of the other features of a microscope. So here it's just going to ask you to do a small test on how you can find the resolution between different threads. Because if you look at that image, you don't know which thread is on top, which one's the second thread, which one's the third thread. So this is an activity that tries to get students to find out, is it the red one that's first, the yellow one that's first, or the blue one that's first? And we only do that through magnification. So if I focus it and then zoom and then focus, which thread do you think is the first one here?
Blue.
Blue. So let's change the order to blue. And what about the second one?
Red.
Red. Okay, so red. Now let's check to see if our answers were correct. All good. Absolutely. So we've correctly done the resolution so we can identify the different layers. And this is quite an important thing for students because they can see when cells are on top of each other, when we've got different layers of cells, for example. And of course, the final thing in this one is using the other parts of the microscope, i.e. changing the position of the stage. So we want the center of the slide to be in the middle of the objective lens. So I want to try and get this E in the middle because I'm looking directly at the center of the image. So this dial on the right hand side, and lets me control the movement of my axes. So I've got my Y, my up and down. So I'll centralize that. And then I've got my X, my left and right. So I centralize that. So now I'm happy that my image is directly in the center and it's perfect to ready to start view. Just check and they're happy with everything I've done so far. So this is typically one of the virtual lab simulations we'll ask our students to do. And of course, it's one of many. You've got focusing microscopes and we've got chemistry related ones. So hopefully you found that quite useful. So yeah, this is linked to that one. But of course, the other simulations, when we've got chemistry, they'll be linked to chemistry modules. Biochemistry, we've got simulations linked to biochemistry, and of course, even microbiology has its own separate sort of pre-labs. So one of the things we ask students to do and engage with during class or in their self-study before coming to a lab, just so they feel much more ready, much more prepared about what they're doing, particularly if they've never handled a microscope before.